Welcome to ABC tutorial. Today we are going to talk about cell division. Cell division is the process by which cells reproduce themselves. That is, the process whereby a mother cell duplicates into daughter cells. Cell division is needed for the replacement of damaged and worn-out tissues. It is also necessary for the growth of tissues and organs. During cell division, there must be accurate DNA replication in order to prevent genetic mutations and other genetic disorders. The rate of cell divisions are usually controlled in order to prevent uncontrollable proliferation of cells, such as those that occur in cancers. Cell division is usually regulated by activators and inhibitors. Cell cycles are the orderly sequence of events in which somatic cells duplicate its contents and divides into two. Somatic cells contain 23 pairs of chromosomes for a total of 46 chromosomes. One member of each pair is inherited from each parent. The two chromosomes that make up each pair are referred to as homologous pairs. However, spermatozoa and ova contains 23 chromosomes each. The 46 chromosomes number are referred to as diploid number of chromosomes, 2n, while gametes are said to have haploid number of chromosomes, n. The cell cycle can be broadly divided into the interphase and the cell division stage called mitosis. The interphase is a state of high metabolic activity. It is during this phase that the cell does most of its growing. The interphase consists of three phases: the G1, the S, and the G2 phase. S stands for synthesis of DNA. During the G phase, there is no activity related to DNA duplication, and they are thought of as gaps or interruptions in DNA duplication. Following the G2 phase is the M phase, or the cell division phase. The G1 phase is the interval between the mitotic phase and the S phase. It lasts for about 8 hours. During the G1 phase, the cell is metabolically active and it replicates most of its organelle, but DNA replication does not take place. Once the cell enters the S phase, it is committed to go through the rest of the cell cycle. For the cell to move from the G1 phase to the S phase, it must pass through the G1S checkpoint. It is at this checkpoint that the quality of DNA replication are being checked. The S phase, which is the interval between the G1 and the G2, lasts for about 8 hours. It is at this stage that DNA replication occurs. The G2 phase lasts for about 4 to 6 hours. At this stage, cell growth continues. Enzymes and other proteins are synthesized in preparation for cell division. Before the cell moves from the G2 phase to the M phase, it has to pass through the G2M checkpoint. Some cells remain in the G1 phase for a very long time. Perhaps they are destined never to divide again. They are said to be in the G0 phase. Most nerve cells, which are non-dividing cells, are in the G0 phase. The mitotic phase, or the M phase, is the stage of cell division in which the mother cell divides into the daughter cells. In mitosis, there is formation of two identical daughter cells. It can be broken down into nuclear division, referred to as mitosis, and cytoplasmic division, referred to as cytokinesis. It results in the formation of daughter cells with diploid number of chromosomes. It occurs in all somatic cells, and it can be broken down into stages, which are the prophase, the metaphase, the anaphase, the telophase, and cytokinesis. In the prophase stage, the chromosomes condense and duplicate into two sister chromatids. The nucleus disappears and the nuclear membrane breaks down. During the metaphase, the microtubules of the mitotic spindle align the centromeres of the sister chromatids at the center of the mitotic spindle. That is, the chromosomes align at the center of the cell. This plane of alignment is called the metaphase plate, or the equatorial plane. During anaphase, the sister chromatids separate and they begin to move towards the opposite poles of the cell. When the sister chromatids are separated, they are referred to as chromosomes. Telophase is the final stage of mitosis, or nuclear division. Telophase begins after chromosomal movement stops. The nucleoli and the nuclear membrane reappears at this stage. Cytokinesis is the division of the cytoplasm into two. In summary, the sequence of events that happen in mitosis are as follows: the G1 phase to S phase to the G2 phase to mitosis and finally cytokinesis. Meiosis is a type of cell division that occurs in gametocytes. It can be divided into meiosis 1 and meiosis 2. Meiosis 2 is essentially similar to mitosis. It results in the formation of 4 daughter cells with haploid number of chromosomes. That is, the daughter cells contains half the number of chromosomes as the mother cells. Crossing over and exchange of genetic materials usually occurs in meiosis, especially in the prophase stage of meiosis 1. In meiosis 1, the chromosomes separate, and this can be broken down into the following phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Crossing over occurs in prophase 1. Meiosis 2, which is essentially similar to mitosis, involves the separation of sister chromatids, and it can be broken down into prophase 2, metaphase 2, anaphase 2, and telophase 2. The differences between mitosis and meiosis can be summarized below. Mitotic division occurs in somatic cell, while meiotic divisions occurs in gametocytes. Mitosis involves only one stage, while meiosis involves two different stages of cell division. Mitosis results in formation of 2 daughter cells, while meiosis results in formation of 4 daughter cells. The daughter cells have 46 chromosomes in mitosis, while they have 23 chromosomes or haploid number of chromosomes in meiosis. There is no crossing over or exchange of genetic materials in mitosis, while there is crossing over and exchange of genetic materials in meiosis.